Hello, I'm Sarah, and I'm the owner of a 15-3 Appaloosa horse who's tested positive for the genetic disease PSSM type 1. You may have heard of this disease. You may suspect your horse has some issues, or someone else's horse, which might indicate you're dealing with an underlying problem like PSSM, or you may just be curious. Whatever the reason, I hope you'll get lots of information from this presentation to help you when you come into contact with a horse with this issue. Maybe you can help another horse and their owner out. As far as I can see, the more we understand, the better we can help our horses and their owners. So what is PSSM? The letters stand for polysaccharide storage myopathy. Polysaccharide storage myopathy, or as I like to call it, something else. Okay, so let's unpack this a bit to start us off into our journey. There's quite a lot of biology here. Most of my presentations will have lots of biology in it, but stick with it. I'll try and make it as simple as I can. But we really do need to understand it so that we can understand what's going on and why some things do and don't work. So the first bit, polysaccharide. What is that? Well, in the body, a polysaccharide is a complex chemical with a specific structure, which is made up of saccharide units joined together. So poly meaning many, saccharide units. And a saccharide is just a single unit of sugar. And there are different kinds, which you may have heard of, such as glucose or fructose, which is the one found in fruit. Maybe if you make jam, you might be aware of that. Now, glucose, let's have a look at that. This is because I don't know how to screenshot on video. So this is glucose. This one at the bottom, if you can see it, the glucose is a ring structure with sticky out bits, and round the ring are the basic carbon atoms. And we'll come onto that later. But the one on the top we call a ball and stick model, because the actual atoms, the carbon atoms are black and oxygen is red, and there is a specific convention to it. And the lines in between them, the sticks, because you can make a 3D model of this, helps you to join them together so you can see what these molecules actually look like. And lots of modelling has gone on. It's been going on for generations. And we're pretty certain of all the different types of glucose. Now, as you can see from that, there might be different ways of joining that together to make a polysaccharide. If you were perhaps building with Lego, you can see how you could make different chemicals with it. And that's the point about polysaccharides. We'll come on to that again in a minute. Glucose is the main fuel that we use in our bodies to give us energy. In most living things, from bacteria, as little tinsy, tiny single-celled efforts, to us and our horses. In this form, the sugar, the glucose, dissolves in the water in our bodies. But, and this is a big but, the amount of glucose in the blood must be very carefully managed. Otherwise, the balance of the fluids in the body start to cause problems. So when someone has diabetes, they need to take over the management of sugar in their blood. So if you know someone who's particularly type 1 diabetic, they need to inject insulin to bring down that sugar out of their blood. It's really important. Blood sugar levels must be kept within fairly narrow margins. So that's why someone with diabetes needs to bring the insulin down, needs to introduce insulin to bring the sugar down actually in their blood. So where does the glucose go when it's removed from the blood? So we're keeping in mind PSSM here. The way in which insulin causes sugar to be removed from the bloodstream is to enable the glucose to enter the cells more easily. So it's not flowing around in the bloodstream, it's going to go actually into the cells of the body. So it could be a muscle cell, it could be a heart cell, it could be another organ, it could be some connective tissue, something like that. But all of our body is made up of cells, so there are plenty of them, billions actually. It's just the way that we're organised. So once out of the bloodstream, it's crossed the barrier into the cell, particularly in the liver and muscle cells we're thinking of with PSSM. The glucose is converted to glycogen. Then it can't get out again, because glycogen is another carbohydrate, but it's bigger and more complex, and it's made out of glucose molecules attaching to each other in a branched chain. So this is when we come into contact with the PSSM issue. The type of glycogen in muscle tissues is where our PSSM horses have a problem. So let's have a look at that. Oops. So this is glycogen. It looks like a big furry snowball. And what it is, actually, you can't see very well from the picture, but it is lots and lots of glucose units joined together around a central molecule. And this is what basically the cell does. It takes all that glucose out of the blood where it's going to cause a problem, sticks them all together and makes a branching large complex molecule. Then it can't get back out of the blood to cause a problem with the fluid balances. Okay. Now, down to the root of PSSM. PSSM causes a problem with machinery in the cells, specifically the part that builds up glycogen. There are two ways a unit of glucose can be joined to another in a cell. You can either do it linearly, so you just make a longer and longer chain, or, as we said before, you can make it branch, so it can be attached at an angle. So in our PSSM horses, our current understanding, and these things will change as more research happens, the machinery for making the long chains out of glucose is defective, it's hyperactive, and it works at double the rate of the machinery which makes the branches. This might not sound like a particularly big issue because, after all, more energy getting stored, more glycogen, it's just doing its job. Isn't that okay? The problem comes when it comes to exercise and when we need that glucose again to provide energy to move the muscles. So here's how. If you could compare a picture of normal glycogen and glycogen in a PSSM1 horse, you would see that there are lots more ends in the normal version. In the PSSM horse, the chains are longer with fewer branches, so the whole structure is less compact. It's looser. It's like that big snowball, but not so hard packed. So this is how PSSM used to be diagnosed, by taking some muscle cells and staining them to find out what type of glucose is present and in what quantities. So let's have a quick look at that now. Muscle biopsy from a PSSM1 horse's hind muscle. The stain is periodic acid Schiff or PAS, which stains glycogen purple. So this one is the PSSM horse, and you can see there's a darker stain compared to this one, which is normal. This one, there's a lot more glycogen and it's packed very tightly into those cells. So this machinery, which is building up the glycogen, being hyperactive, is shoving it all into the cells, slightly uncontrolled way, and that's what you get. And that's how we used to diagnose PSSM1. Now we have a genetic test, so that's different. So imagine the horse has finished eating and the level of sugar in the blood is back down to safe levels. We've shoved all the glycogen, built it up, we've shoved all the glucogen from the blood into the cells, and they've happily made it into glycogen. In our PSSM horse, that's acting really quickly, and then we ask the horse to work. So the glucose free in the cells and the surrounding blood vessels, there will always be a little bit there. It doesn't go down to zero. That can be used to provide energy at first. But in humans, that phase lasts about 10 seconds. So we're going to need some more energy quick. So as the horse continues to work, more energy is required, and other chemicals are released to make this happen. So the building up of glycogen basically needs to be reversed. So along comes the machinery, which snaps off the glucose units from glycogen, so they can be moved into the part of the cell that creates energy from glucose, so those muscles can move. Now, because there are less ends, this process is much less efficient, the breaking down and the bringing back of the glucose. So the result is that muscles have no energy source, they can't move easily, and the horse gets cramp. And therein lies the problem in a nutshell. Sore muscles and everything else that goes with it, all those behavioural things, all those little quirks that your horse might have, all those issues, all the cost, all the vet's bills, that's what is happening. Basically, it's cramp because the horse has no energy source. And of course, the horse can't say ouch. He can't tell you that he's hurting. This is why you get all these behavioural problems. They're expressing pain and discomfort. So it doesn't mean that your horse is mad, bad, lazy, or mean. It means he's got PSSM1, though it can feel like all of those things at times. Those of us who own these horses will certainly be able to tell you that. Okay, let's leave it there. In the next lecture, we're going to look at the genetic side of PSSM1 and how this results in the glycogen problem. So we're going to talk about inheritance and where this gene comes from and what the mistake in it is. Okay, thanks for listening.